Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Sonogenetics. Sonogenetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate data sets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome back, everyone, to the Genetics Podcast. I'm really excited to be here today with Dr. Travis Hinson, who's a leading physician scientist that works in cardiology, genomics, and precision medicine. He's an associate professor at UConn Health and also a faculty member at JAX Laboratory for genomic medicine. He is directing UConn Health's cardiovascular genetics program. And my understanding, Travis, is you spend a lot of your time in the clinic with patients with inherited heart conditions. And he's doing some amazing work at the intersection of CRISPR, gene therapy, and some of the most challenging inherited cardiovascular conditions today. But there's a lot of hope, I think, on the horizon. So we're going to talk about that today as well. So Travis, thank you so much for joining us.
Well, it's my honor, Patrick. I really appreciate this opportunity.
I want to start with a recent review paper that you wrote, and I'll make sure that we have a link in the show notes, but this is about emerging CRISPR therapies for precision gene editing and how to think about this from the perspective of somebody running a cardiovascular clinic. We have many flavors of gene editing. Some are in the clinic, some are earlier preclinical. It'd be great to get a little bit of a 30,000 foot view from you. If we focus in on CRISPR and gene editing, where are the opportunities today, and what are the things that you're most excited about on the horizon?
Sure, it's a big topic, so I'll try to distill it from my perspective as a cardiology physician scientist, taking care of patients mostly with rare genetic forms of cardiovascular disease. You know, it's been an exciting opportunity to experience the growth of clinical sequencing and patient cohorts, our appreciation of how genetics are impacting diseases, particularly for my own personal experience for cardiovascular disease. Our ability to understand and link genetic variants to these diseases has just exponentially grown. However, one of the things that we've been limited by is the ability to intervene on those variants and those genetic causes that we see in our patients. And I think that's really the basis for the opportunity that CRISPR and other genome editing platforms brings to bear, the capacity to go in, find a variant that you have in a patient, program CRISPR to potentially, at least in theory, go in and snip out the bad variant and put in a normal version of it, provides an opportunity as a physician scientist that we could only dream about or felt like science fiction maybe 10 years ago. So the opportunities really are, boundless. There are obviously limitations. I think the variants we know pretty good about which variants are causing disease, although there is opportunities, particularly in more common complex diseases where we're still discovering them. But for monogenic disease, I think a lot of the variants are mostly discovered, and the capacity to go in and correct them is the opportunity. So where are the areas that I see immediate potential for patients? So I think for cardiovascular disease, I'm a big fan of the work that Verve Therapeutics has done, where they're targeting, high cholesterol regulating pathways. So, maybe the audience is aware of this, but, atherosclerosis, where there's plaque building up in the arteries of the heart and the brain and the complete body is a major issue. And we know a lot about that. We know that how LDL cholesterol that carries cholesterol into the arteries, that's a major risk factor. And Verve therapy is targeting the liver, where the sort of source of the bad cholesterol, the LDL cholesterol, using base editors, for example, where they can deliver with a lipid nanoparticle, a base editor that actually generates a disruption variant and key enzymes of cholesterol, and that seems to already be in humans and is making interesting strides. So that's where we are now.
Yeah.
And there's a lot of growth opportunity, but that's where I see things now. And the heart has been trickier because we have to think about delivery, which is a challenge. We have the capacity, I think, now to engineer variants, but one of the limitations is, for some tissues, particularly outside the liver, it's harder.
Yeah. Yeah, that's an important point. We have a number of delivery vehicles to the liver. So if you can go there for the root cause and fix it, then we've got a lot of tools in the toolbox. If you need to go to the heart, maybe you can talk a little bit about cardiotropic AAV, some of the other methods. What are the possibilities if they're not in the clinic yet? The ones that may be in the near future.
Yeah. So about 5 years ago, at least, we were approached by a company called K Therapeutics, which was developing capsids that are myotropic. And what they were doing, which is really important because AAVs, I was actually at the University of Pennsylvania about 20 years ago doing my undergraduate training, and Jim Wilson was pioneering the use of AAVs to target metabolic diseases. But there were safety issues, right, that really ultimately delayed the progression of that capability. But in the last 5 to 6 years, the capacity to do large scale engineering of AAV capsids has provided an unprecedented opportunity to make AAVs now more cardiotropic than they were ever possible. And that's important because we can then use lower doses of virus to get into the heart, for example, which can overcome a lot of the safety issues that Jim had to deal with, and a lot of people have had limitations. So adeno-associated virus is really probably the leading delivery method to get into the cardiomyocyte. And right now, I think the field's able to get payloads such as CRISPR, genome editing, transgenes, and almost any kind of protein you can imagine into essentially every cardiomyocyte of the heart. And that can be done in model systems like rodents. It can be done in non-human primates. And ultimately, I think that's going to be available for human. So it's a really exciting time, although AAV have their limitations because, as you can imagine, a lot of the patients may have pre-existing immune adaptive or innate immune responses to AAV. And so we also have to work on how we can do that safely from an immunological standpoint and make it more available to more patients. But AAV really right now is the leading delivery platform for cardiomyocyte delivery. In contrast to lipid nanoparticles, I think, where you're targeting liver and other. And lipid nanoparticles, there's a big challenge in using those for the heart because they require they have to get through the endothelial layer, which is not permissive in the heart to nanoparticles at this point. But there's a lot of interest, obviously, in adapting them or building other kinds of methods. But right now, AAV really is the key opportunity.
I want to dive into titin. This is and you'll be able to explain this better than me, but it's an enormous gene, and therefore it has a very big surface area for disruption. So is the leading genetic cause of dilated cardiomyopathy, but it is also hard to package up into AAV and other delivery vectors. You've done a ton of work in this space, and I won't do it justice. It would be great if you could give a little bit of a overview of the challenge and the opportunity that we have when it comes to genetic medicines for titin.
Yeah, so as a cardiologist who focuses on the major disease area it's sort of my baby disease is heart failure. And heart failure can be caused by different things. It can be caused by large heart attacks where a substantial portion of your cardiomyocytes die off because of not getting enough oxygen. But it can very commonly be caused by inheritance of genetic variants. Typically in the past, we thought these people who developed heart failure when we couldn't find blockages in the arteries or valve leaks were due to viruses or just completely idiopathic, or maybe they were drinking a little bit too much alcohol. But in the last 10 to 15 years, we know that genetic variants make up a substantial proportion of those patients, of the cause of their disease. And among the genetic causes of dilated cardiomyopathy, which is the kind of common form of heart failure that we see, are variants in the giant gene titin. And what's really interesting is, although we think of these rare variant diseases as being rare, obviously, although collectively they may be common, in the case of dilated cardiomyopathy, titin variants are about 15 to 20% of those patients. So collectively, it's actually a very common disease affecting hundreds of thousands of people in the U.S. alone, where you have a disruption in one allele of titin. And this has been a big challenge from a therapeutic standpoint, because titin is one of the largest proteins in the body. It's encoded by over 320 exons. It has a molecular weight approaching 4 megadaltons, which is the average protein, for example, is about 35 kilodaltons. So we're talking like 20-fold more size. So, vastly too large for gene replacement strategies, which obviously have a lot of opportunities in the clinic. The average AAV can't even come close to even expressing a tenth of it.
Wow.
So it opens up opportunities where, how can we approach titin? So, our group and others have been very interested in delivering, like short hairpin RNAs or splice switching oligonucleotides, or even thinking about ways to leverage CRISPR to either correct the mutation itself or to use CRISPR to do things like transcriptional activation, things to get around the fact that we can't deliver titin itself for patients. So even though, titin has been identified as being extremely important for heart failure risk, it's really not the initial focus for developing genome editing. However, what we're seeing is a lot more interest from industry because, when we're looking at the market size, the number of patients impacted by titin variants is vast and provides a lot of opportunity to make a big difference for patients. However, like the first generation gene therapy, et cetera, really can't address it.
Can you talk a little bit about the variation within the genetic variation that we see in titin, because it's such a big surface area, there's a lot of ways to break it. And in general, you see a lot of skeletal myopathies. What my understanding is some are more severe, some may be more mild. Could you talk a little bit about the spectrum of disease that we know with this gene?
Yeah, so yeah, obviously, like you mentioned, the vast size of titin makes it prone probably to disruptions. And it turns out that there's a lot of complexity to titin. So there's a longer version of titin that's expressed in skeletal muscle as well as the developing heart. And then as the human heart ages or matures to more of the adult state, titin has a splice switch to a shorter version of it called N2B. And it turns out that that leads to exclusion of exons in one domain called the I band, for example. And so when we do sequencing, we have to not only understand that there's a variant, like for example, the most common type of variant that's pathological is called like a premature truncating type of variant or an insertion deletion type of variant. But this can happen in different exons, and it turns out some of the exons aren't expressed in the adult heart. So we have to not only know, is there a truncating variant? Where is it located? Is this in an exon that's spliced out through development? Is this in an exon that's encoding a protein in the sort of the A band, what we call the A band of the sarcomere, which is where myosin is? Those are thought to be the most pathogenic. So the exon location is really important, it turns out, for understanding pathogenicity. So there's that complexity. There's also the interesting observation that even in relatively healthy appearing individuals, that maybe 1 in 1,000 of those individuals will have one of these pathogenic appearing titin truncation variants. And we're just starting to understand why that may be the case, that some patients can walk around with an apparently healthy heart and have one of these pathological looking variants, versus other patients who have the same type of variant, they have to have a heart transplant at age 20. Some of that we think may be related to gene environment interactions.
Yeah.
For example, a really exciting area where we want to make an impact for patients, like in a disease called peripartum cardiomyopathy. Peripartum cardiomyopathy disease affects obviously women who are either right before they're pregnant, or they're about to deliver their baby, or right after delivery. They develop a cardiomyopathy that looks like dilated cardiomyopathy. Initially, that was always thought to be something acquired. There's something about the pregnancy. Maybe they have high blood pressure during their pregnancy, or they may have other gestational issues. But we know now that actually titin variants make up about 10% of the risk for peripartum cardiomyopathy. So this interaction between the titin variant and the stresses of pregnancy come together. And so, we're starting to understand how these gene environment interactions are playing a role in what was historically thought to be more of an environmental cardiomyopathy. So there's a lot of complexity with titin, and that really, makes it challenging to develop therapies, because when some patients walk around with titin variants that look pathogenic but don't develop disease, how do you sort of think about that in terms of developing a gene therapy? Because you have to be able to predict which of the patients who have the variant is going to go on to cause, cardiomyopathy from those that aren't.
Yes.
A really exciting area of research right now.
Do you have any perspectives on which technology is most likely to deliver the first mainstream clinical success here? I know you've done some work in CRISPR activation. There's also, other flavors of gene editing, like we talked about. What is furthest along and or most likely to succeed? They may not be the same thing.
And precisely for titin?
Yeah, precisely for titin.
Yeah, right. So I think there's a lot of cool areas around titin. So there's obviously we're really interested in, which is essentially engineering a transcription factor that can go into and activate titin. And these transcription factors are small. They can be delivered with AAV. We do both CRISPR. We can program zinc fingers. We can program tail transcription factors. So instead of having to deliver the entire titin itself, you can just deliver a transcription factor that's specific for titin. It goes to the promoter and activates transcription that addresses the haploinsufficiency that we see for titin. There are a lot of other exciting alternative areas. So a really cool program is these tRNAs actually that can, so we work with a company called Tavard Biosciences. There's been a couple other companies that have also emerged in this space where they can address premature truncation codon variants that are about 1 in 4 of the pathogenic mutations for titin. This is where you have like a codon that normally would say encode for arginine, but there's a single nucleotide variant that makes it a stop codon.
Yeah,
a stop codon makes it foreshortened titin protein, and it turns out that obviously that's pathogenic. So what they've done is they've actually engineered tRNAs that can recognize those stop codons that are premature, and instead of stopping the protein and making the short version, they actually put in an amino acid. So that's a really exciting therapeutic opportunity because, again, you can deliver that with an AAV because the tRNA is quite small, and you could potentially address any kind of premature truncation codon variant because probably as long as you lengthen titin to its normal length, you would be therapeutically relevant. And even if you put an arginine or a glutamine or so on and so forth, you can be therapeutic. So that's a really exciting area. And there's a lot of interest in actually skipping exons with splice switching oligonucleotides. So there are exons that are mostly because of their large size are enriched for these truncating variants. And if you can develop an oligonucleotide to splice out that exon, because the exon otherwise would be in frame, you can develop a longer protein as well. So those are kind of the major, I would say, the leading opportunities right now.
Yeah, that's interesting. We had a recent episode where we did a deep dive into DMD, and there's a lot of analogs there. DMD, I think, is the longest gene in the genome, but I think that a microdystrophin or a miniaturized version is probably out of scope for titin, just given its structure, or is that something that is possible as well?
People have thought about the mini titin. It just hasn't. I think the challenges with titin are that, although a lot of the parallel approaches for Duchenne have tried to be applied to titin, the biggest challenge, I think, is that there's a normal copy of titin, and there is the capacity for a poison peptide-like effect. And that's made it challenging to do things like, a mini titin, because you could eventually actually compete with the normal version,
right?
Which is in distinction from like Duchenne, where you actually make no dystrophin. So the thought is that any amount of almost regular dystrophin could be therapeutic,
right? Because
there's no competition with a normal dystrophin. In titin, you've got that heterozygous state where you have a normal and so that becomes a big challenge when you try to apply dystrophin type strategies to titin.
Yeah, really interesting. What other causes of heart failure beyond titin are most interesting to you, and where do you spend the most time?
Yeah, we've been focused on, as a cardiologist, I was driven to study these diseases because I think back to my early training, I thought I would become an engineer, and actually I grew up in Louisiana where there were a lot of chemical engineers and you're applying science for practical reasons. And, I started off thinking I was going to be a chemical engineer and learned the principles of understanding how engines work and pumps work. And ultimately became a cardiologist because it had a lot of the same principles, like the heart's like a pump. The blood vessels are like pipes. There's a very clear impact you can have on patients you can see because their physiology is right in front of you. And so when you come to cardiovascular disease from that approach, we think of the heart as a pump. What's the sort of horsepower generator of the heart? Well, it's a sarcomere. And the sarcomere is the intracellular organelle that houses the myosin and the actins, and that generates the twitch force that ultimately underlies the major function of the heart organ. So we've been very interested not only in titin, which is a big part of the sarcomere, but other proteins that make up the sarcomere, and those are things like myosin heavy chain, myosin binding protein C. Those are kind of the motor proteins, and then even the regulatory proteins around the sarcomere, like the troponin complex, where you're your sarcomere has to sense calcium. It's cycling with each heartbeat and turning that calcium flux into force generation by the actin-myosin interaction. So we're really focused on all the different proteins that make up this sarcomere complex. To try, the thought initially was really, when you have variants in the sarcomere, disrupting it, or you're breaking the sarcomere. However, we know now that actually, just like a car, you can have too much horsepower for the car, you can have too little horsepower. I liken it to like a Goldilocks organelle, where if you have too much force, you actually develop a disease called hypertrophic cardiomyopathy, where the muscle is too strong, the walls become too thick, and that's prone to arrhythmias or abnormal heartbeats. That's prone to heart failure. It's actually the most common cause of sudden death in young athletes when this overly strong heart is playing a role. So, but you can have it too weak, and that's really where the dilated cardiomyopathies come out. So we're very focused on the horsepower generator, and we're tackling the different genetic causes. For example, one really exciting program we have in our lab is studying troponin T2, which is the protein that links the troponin that binds calcium to the actin thin filament, the tropomyosin. And it turns out that it's unlike titin. It's a very small protein, and you can actually express it with any AAV. It competes with the normal version, so with the abnormal version, so you can express even a normal copy and do gene replacement strategies. You can base edit it. You can do a lot of really exciting therapeutic interventions. So that's some of the other flavors of cardiomyopathy causes that we're really interested in.
I'm kind of interested in following this engineering thread because I do these kind of discussions across all kinds of different therapy areas, and the brain is nothing like this. It's it's the Wild West in there, and I'm just curious to understand with the heart, is there somewhere right now where an engineering set of engineering principles break down and it starts to look like, okay, we actually don't really know how this works, or can we really think about the heart, and is our understanding of the heart where you can really apply these engineering principles in a really heavy way all the way down, where you can think about horsepower and strain and some of these principles in the way that we might not really get in other organ systems?
Yeah, that's a good question. I think in many cases the heart is a simple organ that engineering principles can help you gain a lot of information about it. There are areas that are beyond, I think, the engineering principles. For example, one area that comes to mind is cardiac regeneration. Cardiac regeneration, we are understanding why the heart doesn't divide and the processes that are controlling that. Our engineering principles really break down. And I would say, I think that's why, our understanding of cardiac regeneration is lagged, say, just the sarcomere function. And so in the other areas, congenital heart disease, where we're trying to understand the blueprint that makes the heart, and with the interplay of the different transcription factors and their timing and development and the different types of cell types that are coming into the heart. Our understanding of congenital heart disease and its mechanisms, both genetically and otherwise, has really lagged behind. And it isn't really, I think, easily understood by engineering principles. But I think when you're looking at heart failure, where we're either talking about titin or other sarcomere causes, or even heart attacks where you're losing a lot of cells, I really do think from the pathophysiology and the physiological perspective, engineering principles really can help you understand. We're really excited actually with these engineering principles is we think a lot of times that, the heart is perfectly tuned to do its function. But I think there's a lot of opportunity actually to go in and tune it in a different way that can be protective, that can be through changing expression of different sarcomere components, targeting how the mechanics translates into signaling, where we can actually make it even better than it is. So I think, so I think that's more of the engineering side, but it's a really interesting organ to study.
That's super interesting because why would it be perfectly tuned, right? There's so much going on. There's got to be some.
So an example, I have a really interesting project that one of my PhD students is working on, where he's his study is trying to catalog the protein composition changes within a domain of the sarcomere called the Z disc, that is one of the most complex protein interaction hubs of the sarcomere. And there's probably 150 to 200 different proteins that come into the Z disc of the sarcomere. It helps both with structure of the sarcomere, it helps with signaling, connects the mechanics to metabolism and translation. It's a really interesting area. And so what he's been trying to do is to define the molecular components of it, or catalog those proteins that are there, but also not only the normal ones, but what happens when you have heart failure, different types of heart failure, what moves in, what changes. And I remember thinking, one of the things that he always observed was that You get a lot more interactions when you have a pathological state. And he said, well, if it's a pathological state, then that must mean the changes that you're seeing are probably mostly, not good changes because it's associated with disease. However, it turns out that we've been actually using CRISPR or other methods to go in and try to either turn on or turn off those targets that we're seeing. And we're finding actually that you can actually better tune the sarcomere through activating genes similar to what we're seeing through the disease process. So I think there is a lot of opportunity to go in these structural areas of the cell, like the sarcomere, observe the changes that are happening, and actually try to either utilize information to develop new therapeutics, like increasing the protein levels that may be thought to be structural. For example, one protein we found is a shock absorber for the cell. It actually can help the cell be resilient from mechanical stress. And we can actually go above normal levels, and it provides resiliency to the cell in the heart and heart failure. So it's a really interesting area for heart failure and understanding the sarcomere and these sort of really interesting molecular changes that are happening to ultimately impact patients, that have heart failure. So that's a really interesting example.
Yeah, I noticed an announcement from a couple of different groups, but yours at JAX in particular, about a week ago, that is on cardiotoxicity. You have this new ARPA-H funding, and I'm curious to learn about that, because that is not just in heart failure, obviously, but across the board. Everyone developing new therapeutics has to think about what impact might this have on the heart. So I'd love to learn a little bit more about that.
Yeah, so historically, many drugs have failed development because they obviously have toxicity in one way or the other. And one of the most feared toxicities that a drug in development can have is impacting the heart. And it's pretty obvious why that would be the case. I mean, if you affect the electricity of the heart, if your heart stops beating for 10 minutes, you're in a catastrophic situation. So, I think historically, avoiding cardiac toxicity is one of the most important goals you would have in drug development. And I think, there's been limited tools, I think, to ultimately try to identify early which of these compounds in our development will do this. Some of that is because actually we haven't had like heart cells available to test drugs. So, historically, you have this beating pump cell, and if you imagine it, it's living in a three-dimensional organ. It's got a biophysical environment that's more physiological. It's contracting like a pump. And how would you even envision having a cell that could help contain all that information, all that complexity? And traditionally, actually, unlike cancer or other immune cells, where you can go in and take a sample from a blood or take a bit of tissue out and grow it in the lab. For cancer and immune cells, you can do that. For heart cells, they immediately die once they're taken out of the body. Moreover, if you snip a piece out of a patient's heart, there's a lot of obvious complications, even high morbidity and mortality for doing that. So we've been living in the dark ages,
right,
to have a system in place to try to predict what small molecules would be doing to the heart without actually having to just go into a patient and doing it. And that has a high failure rate. So part of our ARPA program, which we call Cardioverse, is to build what we think to be a really exciting platform to be able to predict whether a drug is safe or not. And the way we do that is we actually combine complementary model systems. So the iPS technology, this is the induced pluripotent stem cell technology, allows us to regrow hearts from scratch that are human. We can even do that from patient. We take the iPS from a patient that has potentially a higher risk of having a cardiotoxic event, and that's because these are genetically diverse cells. So we can do that across a swath of, say, 100 different patients, just like you were doing a clinical trial on 100 different people. But you do it in a dish with cardiomyocytes derived from those patients. But that's limited because in cell culture, there's a lot of problems with the cells don't really fully recapitulate the adult heart because they're too immature. Their three-dimensional context is different. So we also have a parallel complementary model where we use genetically diverse mouse models, where we deliver the same drugs to a rodent model that has different genetic diversity, different genetic backgrounds, different genetic propensity, and within an organism where you have a heart talking to a kidney, talking to a liver, talking to a brain, that complexity can never be fully modeled with an in vitro system. And then we take all that information together and we use a digital twin strategy, where ultimately, if we can do enough perturbations and collect enough data, we can predict a drug's potential success or safety before we even have to do any experimental data. And that's the kind of digital twin component. So bringing those three components together, I think, through Cardioverse, we can make a big impact, hopefully, on developing compounds with better likelihood of safety and ultimately efficacy to impact the patients. That's kind of the high level
Yeah.
Cardioverse. And what's cool about ARPA is that the scale and the impact of the kind of programs that they're funding is something that you couldn't otherwise do by a lot of other funding mechanisms. Each of those little modules would be like an NIH grant usually, but with ARPA and their scale and scope, it's a really exciting opportunity to bring together things that weren't otherwise possible.
Yeah, I've been meaning to try to get somebody from ARPA H on the podcast because I think they also have there's Welcome Leap here in the UK where I'm based that take these intentionally very big swings, right? It's not supposed to be incremental. It's supposed to be, like you said, we're in the dark ages in this way, and we need to do something radically different. So I think it's really cool, and I was curious about it because I saw it come up the other day. That
yeah, I think in terms of Cardioverse, I think what makes it interesting for us at Jackson Labs is that we're really a modeling organization. Like we try to make we're historically known as a mouse modeling company. However, like we're understanding a lot of the value of the human models, in vitro, organoid, tissue type models. But what makes this really exciting is we can bring genomics to bear on these models. We can do them in complementary ways. And then, obviously we also have the computational capacity to do the potential in silico digital twin. So, I think this is a really exciting organization because we can bring to bear all these things to actually match up with what ARPA, these sort of big swings that they're trying to do.
How do you balance your time between UConn and JAX? What does that look like from week to week?
So, my clinical role is at the University of Connecticut Health Center's cardiovascular department. So we see, I would say I do a clinic every week where the focus is on patients who are referred from other cardiologists who have what we think are genetic issues. That can be across the inherited cardiomyopathies, that can be in inherited arrhythmia, that can be in things like familial hypercholesterolemia or these really aggressive early onset vascular diseases. And then we use genetics as a lens to understand, their risk profile. And then with our background in, either understanding genetics or variant of function or even the physiology of the heart, we can help formulate a very, I think, patient-centric plan for treatment, for screening, not only for the patient, but also their family. And I think really this is, I think even you started by discussing how this is the next era of prevention. Historically, what we've done for patients is they come to our clinic, we check their lipids, we check their blood pressure, maybe we talk a little bit about family history, and then we kind of put into like a calculator, an algorithm, and decide whether we're going to treat them or lower their cholesterol. However, I think what we're seeing nowadays is genetics is really a major new backbone for prevention. We need to understand not only what they have currently, but what they may develop over time, really empowered by the genetic lens. And so I do that about, again, that clinic I do once a week. And then in the lab, I spend the rest of my time where we're trying to develop new insights into heart failure mechanisms, as well as some of the stuff we're talking about genome editing and, understanding, for example, understanding every type of variant in a gene.
Yep.
So those kind of questions in the lab. So it's a really fun career because I'm able to go to the bench, try to work out new discoveries that's never been seen before, and I can apply it to the patients. Although you only have so much time, so you have to kind of pick and choose where you spend your effort.
Yes. You mentioned genetic testing. We talked a lot about therapeutics, and obviously comprehensive and available genetic testing is a precursor to that in many ways. What is your perspective on where we are as a field in terms of access to genetic testing right now?
Yeah, I think if you look at the United States, collectively, particularly in cardiac disease, like genetics is not obviously frequently available. It's not easy to attain. It's really only focused on tertiary medical centers that are heavily in innovation and research. And so I think it's changing pretty quickly. I see for cardiovascular disease, I'm starting to see the adoption of gene panel testing for kind of the monogenic varieties of cardiovascular disease, like the cardiomyopathies, like there's a TTR associated amyloids. We see a lot of that. But one of the issues that, cardiology as a culture is really practical. So they want to see that genetics, the genetic test leads to a new treatment, leads to an obvious direct impact on the patient.
Yeah.
because think about it, like in heart, where you get a bypass surgery, you have a bad valve, you get the surgeon to take the valve out and you put it back in, and you're back to normal in maybe 6 months. And so our culture is that we want to see those impacts, and so genetics has been a little bit slow to really ultimately identify immediate returns on the investment. But what I'm seeing now, I think, is with new therapies. So there's a lot of new gene-based therapies that are coming out now. A lot of it's through things like amyloidosis, where we know that you can inherit variants in transthyretin, which is a normal protein, but in some people it can kind of aggregate in like nerves and heart. But now that if you have these TTR variants, we can do really innovative gene-targeted therapy. So now everyone who has that flavor of disease in their heart is getting tested, no matter where you are in the United States. So that's kind of the first. But we need more and more of these success stories to catalyze more cardiologists to expand the genetic, particularly gene panel. Now, where do I see it going in the future? I mean, I think like a lot of us in this field, ultimately we're going to be doing whole genome sequencing when you're born. We're going to eventually be moving there. And so I think that's where we're going to eventually go. But right now we're sort of tertiary center gene panel testing. And really that's been driven by, do you have a therapy that can be targeted? Now that's in contrast, obviously, to cancer. Cancer for the last 20 to 30 years has really identified gene precision therapies targeting the underlying genetic or transcriptional profile of your tumor. That's where we're going to go in the next, I'd say, 20 years for cardiovascular. We're really on the coats of cancer, but we're not there yet.
Yeah. Yeah, that makes sense. It seems like neurology, cardiology in particular, both are maybe not at the exact same point in that shift, but at similar points where there are a lot of trials, a lot of growing need for genetic testing, but for good reason in many cases, it's not routinely ordered because it doesn't impact care yet, but there's a catch-22 embedded in that, right? And if you don't have widespread testing, it makes the next generation trials a little bit harder.
That's right. Yeah. Yep. Exactly. That's a great analogy.
What else are you excited about going forward as we close out?
Yeah, I mean, I think one of the areas that we see a lot of growth in is in understanding the genetic basis for the non-monogenic forms of cardiovascular disease. Yeah. And I think that's enabled by a lot of the results from these association studies, like genome-wide association studies, and developing polygenic risk scores. So, one of the things that we see in our clinic is, let's say you come to our clinic with dilated cardiomyopathy and we do a gene panel test. Well, our hit rate for these rare pathogenic variants is maybe 25 to 35%. And some of these patients have both monogenic and polygenic, where you have multiple variants coming together. But you're left really in an unfortunate situation where you can't really give them good advice about how to address both their own risk, their own disease mechanism, and their family. So I think I'm really excited the next 5 to 10 years, where I think understanding the polygenic nature of these particular cardiovascular disease, and also like how we can interface the polygenic risk with environmental factors, and we start to really understand the unknown heritability or unknown risk that we're seeing in our patients, which is, I still think, the majority of what we see in the clinic. So I would love to be able to do take a patient who has we do a gene panel and it's negative. We still know they probably have a genetic component. Doing whole genome sequencing, understanding both rare and common genetic burden, interfacing that with environment. Maybe that would be enabled by things like artificial intelligence, where there's so much information, it's hard for us to predict. So the culmination of all those technologies, so that when I ultimately go back to that patient, I have more value to give them. And I think that would really change, cardiovascular clinical genetics.
Yeah, this comment sparked another question that I had, which was how you think about either index individuals or family members, where let's say you have a pathogenic titin mutation or TNN2 or one of these ones that we've spoken about. What does the toolbox look like today to try to understand if and when somebody will become a symptomatic heart failure patient? Do you start imaging family members? Are there other biomarkers that you can look at, like NT-proBNP or other things? And how do you see that evolving, where maybe you have a 20-year-old family member whose parent had heart failure, and how do we think about that case?
Yeah, that's great. So, I mean, actually, this is how we see our families. There's a one index case, a person who has a more severe version of the disease. We then identify a genetic variant that we think is causative for their disease. And then the question is, what do we do next, right? And what we try to do is the patients really are entry point into their whole family. And we try to say, okay, let's apply this test now that we know what's going on in you to the rest of your family. And it's usually these are autosomal dominant diseases generally. So about 50% of their siblings, their children, their parents will also carry the same genetic variant that they have. And what we do is we work with them to identify a way to see, do they have current problems with their heart? So we'll do imaging. Usually in the case of heart failure, we'll use echocardiogram, which is a non-invasive ultrasound to look at the function and structure of their heart. Many cases we'll see people who didn't know they had any abnormalities in their heart because they've adjusted over many years to their cardiac performance. So we'll identify those people. And the really value to that is if you can identify treatments, even treatments that aren't genetically targeted, but just our bread and butter heart failure treatments, like things like beta blockers that target the neural system, or angiotensin converting enzyme inhibitors that target things secreted by your kidney or blood vessels, that initiating those, even though they're not perfect treatments, if you catch it early, you can prevent the worsening of their heart. The real challenge has been what happens in their kids who may be teenagers or younger children. Do you do the sequencing early with them? Because that can have impacts both on things like athletics, or, those are tricky questions that we're still working on. But ultimately, we do sequence the entire family. We help develop a program where we're trying to identify a screening, usually with imaging, to identify those who may go on to develop the disease and initiate these treatments earlier. And we're dealing at the same time with really great areas like, how do you deal with genetic counseling for younger people who may be doing athletics, or they may be a professional athlete and they have a troponin variant or titin variant. And we know that largely we're kind of living in the dark ages for how do we properly counsel people, with that situation. But I really see this as a family-based care model.
Yeah.
I see one patient, but I'm really thinking about their entire family. It's a new paradigm. Doctors normally think about the sickest. If you think about it, really, it's the opposite of what we normally do, right?
Right.
I usually see patients who have catastrophic heart problems, and their treatment needs to be like replace the valve or do a stent into the artery or bypass artery. And then we ignore their entire family who in the next 10 years could develop the very same problem.
Yes.
Doing nothing for them. So we're saying, let's focus on the person who has these. We learn from them, and then we try to treat their whole family. It's a completely different paradigm in healthcare.
Yeah, I mean, one of the things that's interesting to me about this podcast is I hear this exact same theme across so many therapy areas. I've had a couple of podcasts recently in FTD and ALS where you have the same paradigm. I've told my story a couple of times. We have a genetic kidney disease in my family. It's an amyloidosis, very rare. I'm a gene carrier for it, so I'll probably get a kidney transplant at some point. But and this is no disrespect to any doctors involved. They're all trying to do what they can, but there's just limited things they can do actually for somebody in my situation and somebody who's a GRN carrier of FTD, for example. But the more availability of testing we have, the more it opens up early detection prevention through surveillance with minimally invasive biomarkers or treatment eventually, right?
Yeah, and ultimately we need to do. It's a perfect example, and I think it suggests that it's a public health issue almost, where we need the insurers to be on board. We need government to help play a role. Like, you have the UK Biobank doing all the sequencing. You have some countries that have really invested in genetics as an important public health issue. Like, we need to address this from more than just physicians on the front lines. We need to employ genetics as prevention across all the stakeholders. And I think it's going to happen. It's just, you need more success stories and more investment in this area. But, I'm really excited to help at least be a part of this process. And I think it's going to impact patients on a completely different level and their families. I mean, ultimately, the goal here is to find people who don't have disease yet, who are going to probably develop it, put them in clinical trials or put them on treatments that prevent them. Because we all know if you're early to disease, you can do a lot better job with it. But like, does an insurance company want to pay for someone who doesn't have a symptom yet?
Right.
Do you want to do sequencing in people who are healthy? That kind of concept is different than what we do currently, and it's not a short-term value proposition, but it is a long-term value proposition, right? Because if you're going to develop it, like your example, like maybe you will develop kidney disease in 10, 20 years, who knows? But if you could be treated earlier and prevent it, you're saving obviously quality of life, but you're saving actually even money for the system, but it's in 15 to 20 years.
Yes.
And
Yeah.
you may have changed insurance companies by then, right? Which is opening up a whole different can of worms, but we really should be invested in the individual and the family's health beyond the short term.
Yeah. Yeah, it's a big challenge. I think the newborn screening programs are an interesting Trojan horse into this, because if the health economics work out there, where 1 in 1,000 kids we can treat or prevent something catastrophic through the newborn screening, and then you can bank that DNA for future. I think there is a world where this happens just because it's economically viable in the newborn setting, but that doesn't do anything for those of us who are not newborns today. So we will
right.
the problem separately.
Yeah, I'm very excited for the newborn screening value, and I think it's really, you're right. It's a great demonstration project to motivate more investment in larger scale sequencing. I think it puts on the people who are kind of doing genetic medicine also to be very practical, because we also don't want to overreact to variants that may have very low risk.
Yes.
We have to make sure we actually responsibly react to genetics, because genetics, when you're doing genetic testing, the other thing we haven't talked about is it can create a lot of anxiety, a lot of uncertainty, and a system where, for example, doing more of things it generates more revenue. We don't want to do that. We want to be responsible. We want to be consistent. We want to help patients, but not necessarily do things that are going to generate more tests that aren't necessary. So it's a two-headed monster.
Yes. Yeah, I couldn't agree more. And I think that there is going to be, it's going to be interesting what we learn about the prevalence, the prevalence, but also the penetrance of some of these genetic variants when we are sequencing in a way that's completely non-ascertained compared to sequencing in families, where you can get a really biased picture of how penetrant is this. When you have an index case in the family, you're conditioning on background polygenic risk and lots of other things that we see time and time again with bigger population genomics programs that we're revising our penetrance estimates down. So I think we'll see that more with newborns.
Well, and I think that's we see that in heart obviously as well. And I think it gives patients a lot of positive feeling about the fact that if there are patients who have this similar genetic issue, but they do well, it empowers them that there are choices and there's opportunities to go beyond what you're born with, to make a difference, and you're beyond doing a treatment, but you could actually make a difference for your own risk. And I think that's a positive result, and even enables the importance of genetics as well. Because it can sometimes, I see this frequently in a patient, you find a genetic factor, they don't have significant disease yet. They're asking, well, Dr. Hinson, can I do more exercise? What about my nutrition? What about my weight? Can I now I'm inspired that I have this factor and I can now make a difference with good decisions. And that's equally important in genetic medicine.
Absolutely. Travis, this was a super episode. Thank you. I learned a ton, and I really appreciate you taking the time.
Thank you. It's been an honor, Patrick. I really appreciate it.
And everybody, thank you for listening. As always, we'll see you next time. Thanks, as always, for tuning into the Genetics Podcast. If you enjoyed today's conversation, the best way you can support the show is by sharing it with a friend or colleague who might find it interesting as well. We'd also really appreciate if you could subscribe, rate, and review us on Apple Podcasts, Spotify, or wherever you listen to podcasts. This helps other people discover the show when they're searching for content on genetics, precision medicine, or biotech. We always want to hear from you as well. If you have feedback or questions or want to be featured on the show, you can email us at podcast@sonogenetics.com. You can find me on LinkedIn, Patrick Short. You can find us as well, Sonogenetics, on LinkedIn, or reach out on Instagram at The Genetics Podcast. And finally, a special thank you to the team behind the show who makes this possible. Joy Ismail produces and manages the show. Jane Reddy-Webster keeps us organized, and James Pierce from Selective Frequencies for his expert audio engineering. I'm Patrick Short, your host. Thanks again for listening, and we'll see you next time on the Genetics Podcast.